Right, I believe we are more or less connected. So let's start with this second sRedList webinar. The first one was this morning at 9 a.m. Central European time, and now we have a second webinar to accommodate for a few more time zones. My name is Moreno Di Marco from Sapienza University of Rome, and I am one of the PI of this initiative together with Luca Santini. And I'm going to spend just 10 minutes to introduce you the sRedList platform for rapid and effective global biodiversity monitoring. After a quick introduction by myself, I will leave you to the main event, of course, which is going to be the presentation by the working group postdoc Victor Cazalis, who is the main person behind the development of the platform, and also the person that is going to be able to tell you more of how it works. So the rationale that led us to start this work is something that we're all familiar with, and that's, of course, the need to improve, support, and expand the IUCN REDList. We know that REDList has been expanding in a very rapid way, especially in recent decades. There is more than 150,000 species now, and that's, a globally hugely relevant tool and globally important tool to track change in extinction risk over time. But at the same time, as the tool expanded, we have also seen a few problems that characterizes the use of this REDList. So, for example, we know that there still is a taxonomic bias in the list, for example, with the relatively little representation of invertebrates compared to vertebrates. And we know that even for the species that are represented in the list, we have at least a couple of important problems. The first one is many species are data deficient, meaning that we don't know what their threat status is. And the other one is a good amount of species, roughly 15 to 20%, depending on where you look at the REDList, are considered outdated. So their last assessment is older than 10 years, meaning that their current risk of extinction might be not represented anymore by that category. The reason for those challenges is that the information that leads us to assess species into a category of risk is fairly complex. We need to know information about species distribution and occurrence and its abundance and the trends over time. And let's be honest, most of the time those data don't really exist. And so REDList experts and assessors do the best they can with available information, but sometimes this information is insufficient. For most species, I would argue, this information is sufficient to come up with a truly complete assessment. And that's without considering the need for periodic reassessments. Now, we all know how time-consuming and resource-consuming it is to run periodic reassessments of entire group of species. I think the latest one published a few days ago was the Global Amphibian Assessment. And let's just think of how long it took to complete that effort, and let's scale that to every group which needs periodic reassessment in the REDList. So it's no wonder that we are dealing with the problem of keeping the REDList up to date. At the same time, even for species that are assessed and are in the list, there is often inconsistency. There is inconsistency in which criteria can be applied to which group. In theory, every species is supposed to be assessed against every criterion, and then whichever criterion gives you the highest category of risk is the one that will ultimately determine the category of the species. But to be honest, a lot of the times, not every criterion can be applied to every species. There is groups of species where you can only apply one, maybe two criterion. That generates an inconsistency in the available knowledge and in the representation of this knowledge in the listing, because some species will have a more complete and accurate assessment, and some other species will only have a more limited assessment, mainly based on one, perhaps two criteria. At the same time, the application of each and every of those criteria require knowledge of parameters that are sometimes available and estimated, sometimes just guess estimated based on expert knowledge. And that also generates inconsistency on how, different species abundances or area of occupancy or area of habitat or range sites and so on and so forth get evaluated by experts of different groups. Now, the possibility of introducing automated tool or even statistical framework to assess species extinction risk was discussed several times. One of the first paper presenting the possibility of predicting extinction risk based on REDList information was one from Andy Parvis and colleagues in the year 2000, and they essentially said, perhaps extinction risk can be modeled, perhaps there is automated tools to determine species extinction risk. and about 50 papers were published on that topic in the following 12 years, and virtually none of them ever really informed the red list assessment. And actually, in 2012, this other paper came out that says, are those studies of extinction risk really useful for conservation at all? And then another 10 years comes by and another roughly 50 articles on the same topic gets published, and still the uptake of this information in the red list is very, very limited. And so last year we published this paper where we realized that the main challenge is not to come up with better or improved statistical tools to assess species extinction risk, but the main challenge is actually bridging the research implementation gap in the UCN red list assessment. So making sure that scientific advances in extinction risk modeling and automated methodologies really get to communicate with the actual application of those tools in the red list. And this is where we essentially realized that the reason for this misalignment, the reason why, the scientific efforts and all those papers were pretty much detached from any practical red list application, was a misalignment in interest, a misalignment in output. So red list assessors really have good knowledge, strong, deep knowledge about the red list work, which are the guidelines, how those guidelines apply to their specific species of interest, how each parameter can be estimated in a way which is relevant for that particular species. And on the other end, data scientists or modelers, those, and I'll put myself into that category perhaps, those of us who have been publishing those papers I was talking about, they have modeling expertise, and they typically apply those techniques to many species in a more or less generalized way. We don't necessarily have the knowledge to apply those modeling approaches to individual species in a way that makes deeply sense for the biology of that species. And there is a divergence in interest. There is divergence in outputs, and really the only way to merge and to really recover from this research implementation gap is to improve communication between those communities and to co-construct tools between these communities, making sure that whatever is produced by scientists really starts with a purpose, which is going to have a direct application in the red list. This is the rationale behind the creation of a working group, which is called the sRedList Working Group, which I have done together with Luca Santini, and that involved roughly 30 people from different academic institutions, non-governmental organizations, governmental bodies, and several people involved in a direct way with the UCL red list, and also technical modelers and developers of IT infrastructure facilities from LifeWatch, the LifeWatch team in Italy, that really helped us to co-create and co-design a tool that can be used by red list assessor, as being co-designed by red list people and red list assessor, and is freely and easily accessible to everyone, and makes available automated techniques to perform red list assessment in a way which is systematic, standardized, controlled, and rapid. The goal of the sRedList working group is fairly simple if you think about it. So it's essentially to facilitate, speed up, and make red list assessment more consistent. And we want to do that to reduce the taxonomic bias that characterizes the red list, to keep the red list up to date, so to overcome that outdated problem that I was mentioning before, and also to guide reassessment. 150,000 species is a lot, and not all of them are going to change their status. In fact, a very small percentage of species change status between one assessment and the following one. So, perhaps we can identify priorities and which of those species are more likely in need of a reassessment. So the tool, the sRedList working tool and platform we're gonna hear about today, really can do all those things, can help you with assessment, can help you with reassessment, can help you understand which species is more of a priority for reassessment because it's more likely to change its conservation status, and also how to deal with the limited knowledge associated with data deficient species, and perhaps some of those species can be actually reassessed into a category of risk. The platform works into sequential steps, and Victor Cazalis, who is the mind behind the creation of this platform, will give you all the details in a minute. But as a general overview, the platform really asks you a set of questions and information, and is completely customizable based on your experience and your knowledge of a certain species. So it will let you decide whether you want to do a first assessment of a new species or a reassessment, for example. Then it will allow you to define the distribution of that species, which could be as simple as saying, I just want to take the available red list polygon from the UCN red list website, up all the way up to the opposite direction, which is I want to create a new map for that species starting from a set of occurrence points and polygons, for example. And once the distribution is created, you can estimate a lot of other parameters that can be used for criterion A and criterion B and C. You can, for example, verify in which country the species is present, what's the extent of occurrence, what's the area of habitat, how to convert the area of habitat into an area of occupancy for that particular species. You can use that information to estimate trends over time. You can estimate trends in area of habitat. You can even convert for a few species for which, a few groups, several groups actually, for which we have information available. You can even convert that trend of area of habitat into a trend of abundance, for example. In doing that, you can automatically estimate level of fragmentation based on REDList guidelines of fragmentation. Then you can measure trends in species suitable habitat based on remote sensor product. All of this can be done in a customized but automatized way, in a way which will make every assessment technically comparable with each other. At the end, each and every of the choice you make will be calibrated and can be edited. So each and every information you want to use to determine the area of habitat, the area of occupancy, and everything else can be edited and customized, and we will keep track of your choices. And what happened at the end is that the platform will produce a file which is directly compatible with the SIS system. So via the SIS Connect, you can take the file produced by the platform with all the characteristics and all the information and all the settings in its memory, and you can automatically enter that file into the SIS. And at that point, you can treat your assessment just as any other assessment which is included into SIS. The platform is going to be on live tomorrow, and we really hope that many of you will try this platform and will use it for their assessment. What I can anticipate is that we ran 3 tests before releasing the platform with subgroups of REDList assessors that were experts of different parts. I think we have tested plants, fungi, insects, and a few vertebrates. And what we found out is that there is a little bit of a small learning curve that doesn't last long. I'm talking about a few hours, maybe a couple of days. But then in the longer run, every assessor came back to us and said, yes, if you have to do more than one assessment, this is going to help you. This is going to make you save a lot of time. But from our point of view, this is important. Saving your time in the long run means you can do of course, assessment with less, in a way which is less time consuming. But importantly, I think we are also improving consistency here, because everything which is done in an assessment now can be tracked. And so you can start really compare the extinction risk of different groups if both groups have used this platform. I don't want to take a lot more of your time. I'm just saying that what you will see today is the beginning, hopefully. We have plans to expand the platform to make it include additional functionalities. For example, we are currently developing modules to improve the assessment of data deficient species, understanding which of those species will likely become this data sufficient based on some scientific publication we recently produced. And we also will implement an entire module on calculating climate risk in a way which reflects the REDList guideline and can be directly applied into criterion A3 on future population trends. We don't have enough funding at the moment to do this model and these modules, but they're pretty much in our pipeline, and we hope we will be able to secure those tools in the near future. So with that said, I will leave the floor to Victor Cazalis, who has been the creator of the sRedList platform. And once again, I really need to thank all the working group for the brilliant work they have done in just 3 years, which will led us to this point. Thanks very much.
Thank you, Moreno. I will share my screen. Moreno, can you confirm you see it?
Yes. Yes.
Okay, thanks. Hi, everyone. It's a great pleasure to be able to present you the sRedList platform today. I'm Victor. As Moreno said, I was part of the team who is developing the platform. Currently, I'm in charge of some maintenance and some development of the platform, and I'm probably your best contact point if you want to ask questions. Feel free to write me. Today, I will present you the platform in 3 parts. First, I will show you the documentation and help that we created to help you navigate the platform and understand how to use it. Second, I will pretend that I'm an expert of ant species from the Amazon and show you a full process of what you can do on the platform and what information you can gain there. And third, I will explain you how you can use that information in your assessment in SIS. So let's start with the documentation. Here is the homepage of the platform, and you will see on the assessment page that your best friend will be this little eye icon that you can find everywhere on the platform, and that will make appear a tooltip that gives you some text of what this step will be and how to choose the different parameters at that step. On the homepage, you have access to the platform documentation. That is a full PDF with a lot of text that is exactly the same text than the one you can find with the tooltips I just showed you. So for instance, at the EOO analysis, you can read that text to understand what we do. Another document that we have is a cheat sheet. So it's a 2-page document that presents you the main steps, each parameter that has to be defined at that step, and a few details here, and also the output that you can get out of that step. It's not complete, like you won't have all the details here, but it's maybe something to print on the side if you're doing a lot of assessment with the platform so that you can remind yourself what's the next step. And we also have 13 video tutorials that you can find here. Each of them presents one step of the platform, one main step, in quite a lot of detail, much more detail than what I will show you today. So here you will have every parameters described. Finally, we have a forum where you can register and ask questions, report some stuff that are not working, make suggestions, and so on. And we can also ask you questions. So I will monitor the forum and be there to answer your question if you have any. You can also write directly to contact us on the footer of the platform in order to send us an email if you prefer. Okay, that's it for the documentation. Now I will show you a full assessment, a first assessment of an ant species that I chose, that is Azteca xanthozoa. So the first step is to spell the name correctly, of course. And then, as Moreno was saying, we need a distribution to apply to perform all the analysis on the platform. So the first step is to select that distribution. If a distribution was already on the platform, it would be suggested here. But for this species, there is none. You can upload a distribution if you created yourself a distribution in GIS. You can upload the shapefile here. And in that case, I want to show you how to create a distribution from occurrence records on the platform. So you see here that on the top of each page, you have a little camera that brings you to the video tutorial of that step. To create a distribution range map, we first need to gather some occurrence records, then to filter the ones we don't want to use, and then to create a polygon distribution range map out of that. So the first step is to select where to get the occurrence records from. I want to use the records published on GBIF. And also I gathered some records from the literature that are not yet in GBIF, also my own observations if they are not there. So I want to use them as well. Then I will run the first step that will download the records in GBIF and my records and will merge them together. It will return a map. You can see on that map the distribution of those points and those coming from GBIF and those uploaded. And then we get to the page, to the step to filter those records in case we don't want to trust some of them. At that step, you have an interactive map where you can zoom in, click on the different occurrence records, and get a link to the GBIF webpage of that record. You see that some of them are in purple. This means that they are considered not valid based on the filtering steps parameters that we designed. Why is that? This one is because of coordinate uncertainty. So here, I said that I want to remove every point with more than 10 kilometer uncertainty. And this one has almost 300, meaning it could be anywhere around here. So I want to exclude it. This one is also purple because it was made in 1895, which is before 1900. So by default, we remove every point before 1900. If you want to remove every point before 2000, you can also do that. Now, if we zoom out a bit, we see that there are some weird points coming from GBIF, one in the Netherlands, one in Brazil. Those two are clearly wrong. They are in naturalist institutions, so they are clearly the wrong coordinates. So I don't want them. This one is a bit less clear. It's in Santa Marta, and the observation was made in Santa Marta. I went to check the link before, but it's very old, almost a century old. So maybe there was a taxonomical mismatch or something like that. So I don't want to include it either. So what I will do is to filter by geographic extent. Basically, I want to remove every point that has a higher longitude than here. And to define that, I can check. Here you see the longitude and latitude of my mouse. And here you can see that it's about minus 76. So I will say that the maximum longitude of my point is minus 76 and run again step 2. Now you see that they are excluded, and the reason why they are flagged is it's outside the extent that you set here. Okay, now I'm quite happy with my point, but when I zoom in, I see that this point is made at sea, which I don't trust because my species is an ant, purely terrestrial. So I can also ask the platform to exclude every point made at sea, which is done now. So now I'm quite happy with my set of occurrence records. So I will go to the next step, which is to create a polygon range map. I won't detail everything you can do here. There are a lot of possibilities. I will just, and you have the details in the video tutorial. There is a specific one about that step. And I will just show you the alpha hull that connects your point a bit as the minimum convex polygon, but allowing some concave incursions within the points. So you see here that we got close to the point, while a minimum convex polygon would have drawn a sharp line, a straight line. But here there is a big part of the range that doesn't have any distribution and was not, that didn't have any occurrence record, but was not excluded from the range. And I would like my range to go like that. So what I will do is that I will adjust the alpha hull parameter, and I will decrease it. That way it will be closer to the point. I can try 0.7 and see what it does. Okay, now it's quite good. I like the global shape. The only thing missing is that now you have only the points. So the points are at the edge of the distribution, which is not true. Probably the species is also present beyond those points. It's just that it's quite poorly sampled. So what I will do is that I will buffer a bit around that distribution. For instance, I can do 100 kilometers, and as I said, I have a purely terrestrial species, so I don't want to keep any sea parts in my distribution. And I get this map that starts to look like RedList distribution, and I think it's quite good. The only thing is that you have some sharp angles, for instance here. Also here, it's not very elegant, let's say. So the last step is to smooth, if you want, that distribution between 0 and 100. I will see what 50 does and adjust based on that. Okay, now I think it's quite good. Here you have a little rounded thing, here as well, here as well. So I will use that distribution in the next analysis. At the end of the process on the sRedList platform, you will get a zip file that will include the distribution as a shapefile. If you are super happy with the distribution we provided, you can use that in your assessment. If you are half happy, you can take it in a GIS software, edit a bit, remove an island, change a bit a curve, and so on, and use it in your assessment. And if you're not happy, you can just dismiss it. Also, we have occurrence records in the final output in the shape that should be submitted with your assessment. So that's a compatible file with the assessment. You may want to check it manually, but it includes all the valid occurrence records that were gathered on the platform. Okay, now we have that, we can go to step 2, which is to extract countries of occurrence. Every step on the platform, except the first one of the distribution, can be skipped. You can just go next if you don't want to extract countries of occurrence, if it's not useful to you. In that case, I will show you quite everything. So the countries of occurrence, we use a map that has the exact same country names and subnational entities than the one you can find in SIS. So the idea is that based on the distribution, we will tell you which countries of occurrence overlap with your species. You can click, so for instance, here you see that Panama, the species is present, that in Mexico, the species is present, including in the subnational entity of Oaxaca. And you see the different occurrence records. With this little list icon, you can access a text result of that, where you will get the list of all countries of occurrence, including subnational entities. So for instance, in Mexico, we have those different subnational entities. You can see that some of them, for instance, El Salvador, is in italics. That means that we think the species is present there because it overlaps with the distribution, but we don't have any occurrence records to confirm that. So it will be coded in the final output as possibly extant. You see here is El Salvador. We have distribution of the species that overlaps, but no occurrence records. So in the final output, you have countries.csv that includes all those countries of occurrence, and El Salvador will be coded possibly extant, while the Panama, for instance, will be extant. Also, when we extract countries of occurrence, we extract the realms, here neotropical, and you can see, you can click realms to make appear the different borders between the realms. So here we see that we are clearly in neotropical and not in the next one. Okay, the third step is very easy. We calculate the external occurrence, mapping the minimum convex polygon around the distribution, and we calculate the value. You can click on interactive map if you want to look more detail on that. Then we reach a bigger step. Currently, this step only makes sense for terrestrial species because only terrestrial area of habitat methods have been published. But in the future, people are working on developing something similar for freshwater and marine species. So we hope that we can integrate that later into the platform. The idea of that step is to help you calculate the area of occupancy. And what we will ask you is habitat preferences of the species and elevation preferences of the species. And based on that, we will use some global maps of land cover and elevation to try to map the suitable habitat of your species. If the species is already on the red list, the habitat preferences will be automatically loaded. This one is not yet in the red list. So you can either select it manually, or you can use that button to extract the habitat of the occurrence record. So we look at the occurrence records gathered in the first step, and we tell you in which habitat they fall into based on Young et al. Global maps. So here you see that this species, the occurrence record of that species, fall mainly in 1.6 IUCN habitats, so some tropical forest, and 1.9, which is tropical mountain moss forest, and also a bit in 14.1, so arable land. What I will do is that I will say that I'm sure 1.6 and 1.9 are suitable, and for 14.1, I'm not sure, so I will put it as an unknown suitability, and the platform will calculate area of habitat with and without that habitat preference. For elevation preferences, it's the same. If it's in the red list from a previous assessment, we provide it to you. And if it's not, we calculate the minimum and maximum elevation within the range that we draw together in the first step. So in the range of our species, elevation goes from 1 meter to 5,000 meters. I'm pretty sure my ant doesn't go up to 5,000, so I will check where the occurrence records fall. And the maximum elevation of the occurrence records that we gathered is about 1,700. It doesn't mean that it's the upper limit of my species, but it's clearly not 5,000. So what I will do is that I will say the minimum is 0, that makes sense, and the maximum may be 1700, but I want to check if it was 2500, would it make a big difference? Then I will add that, run, and explain it to you while it's running. Based on the area of habitat, we can also estimate population density. So here I say that I think my species has a density between 10 and 20 mature individuals per square kilometer. I'm not an ant expert. I hope it's not shocking. And based on that, we will calculate the possible population size if all the suitable habitat is occupied by the species with that density. While it's running, I should warn you that the maps we use, the global land cover maps from the ESA CCI, are useful but not perfect. So in that step particularly, but in every step on the platform, we try to provide you the best data, best analysis possible, but it will sometimes not totally match what you expect for your species. So every result of the platform, you should judge with a critical eye and decide if you use it or not, or if you edit it a bit. The first result you will get, if you started from occurrence records as we did, is a map of the known area of occupancy based on occurrence records. So I will zoom in so that we see a bit better. It's basically what you already have in GeoCat, where we take the occurrence records and draw 2 by 2 kilometer grid cells around, and we sum those grid cells to get an estimate of the area of occupancy. In that case, the lower bound based on occurrences is 344 square kilometers. When you look at the map, you clearly see that the area of occupancy of the species is not 344. It's just that it's quite poorly sampled across a big range, but that the area of occupancy is likely much bigger. And to get that much bigger upper bound, we use the area of habitat. The idea is that we tell you, based on the habitat preferences and elevation preferences of your species, where we might find suitable habitat. That's in dark green here. And the light green is, it would be suitable if 14.1 habitat is suitable, and if the upper limit of elevation is 2500, so like the maximum area of habitat with the uncertainty you provided. And then we rescale that map at a 2 by 2 kilometer resolution in order to calculate the maximum area of occupancy. So that would be the area of occupancy of your species if every suitable habitat is occupied. And it would be closer to 500,000, so quite far from the 344. Then the idea is that you use the map and your knowledge of the species and the region to estimate what is the most likely between those two estimates. And maybe you will know that the species is not present there, and you can retract that from the area of occupancy. Also, we estimate population size, as I said, by multiplying the area of habitat by the population density that you provided. Based on the exact same methodology, you can calculate trends in area of habitat. So we map area of habitat in 2020 and 10 years or 3 generation lengths before. And we'll provide you a map and an estimate of the trend. As I said, it's based on the data that is available, and it's quite conservative. It's not because it tells you that the habitat didn't decline, that the species didn't decline, or that the quality of the habitat didn't decline. It's just the ESA CCI doesn't allow you to show a decline in area of habitat. I don't have time to show you this step today, but I will show you another way to calculate trends in habitat for forest specialists. The next step possible is to map fragmentation of your habitat in order to assess the severe fragmentation of your species. I won't have time to show you this one either, but you can click here to see the video tutorial and access that step. The one I wanted to show you is that we allow to calculate trends in some global products. So either forest cover, human modification index, or human density, and probably we will add some in the future. Forest cover, we use the global forest change map by Hansen et al. And we calculate the forest cover in 2022, which you can see here on the left, and the change in the last 10 years or 3 generations, which you can see on the right. So it helps you to see where there was deforestation within the range of your species, and also to get an estimate of the relative trend in the range. So now you see that the forest declined by 9% in the last decade, which you can use to inform criterion A2. Finally, you reach a step that summarizes everything that was gathered on the platform. First, the taxonomy of your species, which is only important if you want to use SIS Connect, which I will talk about in a second. And then the different parameters that were gathered in the platform. So for instance, the extent of occurrence, and we provide a standard justification that you can and probably should always edit and complete with some other information. You can also update the estimates. For instance, the area of occupancy. I told you that I don't trust this 344. So I will say that it's at least, at the very, very least 2000, so that it doesn't qualify for threatened. And I should go in there and edit as well the justification. We also have population size and some other. And I will add the information from the forest cover change. So here you should write something convincing, blah, 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 for the justification that I want to use for criterion A2. It's inferred based on quality of habitat, it's not reversible, it's understood, and it's not seized. So here you can add many parameters that are in SIS. You don't have to. Basically, every assessment, you will still have to go to SIS to edit and complete your assessment. So you can fill it here if you want to save some time from SIS, or you can just ignore that and then finish your assessment in SIS. In that case, I just want to say that the species is probably declining, and no, I don't know future trends, so I will not fill that. Then you can assign category. It's just something to give you a visual conclusion of the platform. So we will show you, based on the information that you filled, which criteria are triggered, but the real criteria calculator will always be on SIS. Okay, so here you have the different criteria and the category that is met according to the information you filled and completed on the platform. Here the species looks like it's the least concern or near threatened. And now we reach the end of the process on the sRedList platform, and you can download the zip file that I was talking to you about. That will include the distribution files that I was mentioning, also some files like the countries of occurrence, the habitats, and some files that include the results. So for instance, here you have all parameters and justifications that you completed in the final step. And also an important file that is the sRedList report. It's an HTML file that you can open in a web browser, and that gathers everything you did on the platform. So every choice you made, for instance, it reminds me that I chose to remove every point with higher latitude than -76. It gives me the interactive map, the different histograms and results that I had on the platform, the list of countries of occurrence, and so on. And at the end, a table of the output that I edited in the summary page. This file is here to remind you what you did, but also to use in order to inform your assessment and communicate with other assessors and maybe reviewers of your assessment to get more detail on what was done on the platform and what was the results. I didn't show you everything that is possible on the platform. Just to give you a quick overview, we also enable, wait a minute, I need my notes. We also enable to map freshwater distribution using hydrobasins with different possibilities. We also enable to extract countries of occurrence for marine species using the exclusive economic zones. We also enable to crop by country. If you're working for a national red list assessment, you can say, I'm just interested in the part of the distribution that falls in Greece, for instance, and you will do the whole analysis only for the part of the distribution that is in Greece. Also, we enable to calculate fragmentation as I was saying. Now, let's move to the last part, which is how to use that in your SIS assessment. So I have a small visualization problem with Zoom. Okay, I will go like that. So the first most easy way probably to, did I close? Sorry. The first way to use that information in SIS is to just select the information that is useful and copy paste it to SIS. For instance, if you like the area of occupancy estimation, you can just go there, go in AOO and copy paste that information and also copy paste the justification. The idea is you will just take the information that is useful to you. So maybe some estimate, maybe you can also use the map and that text to decide what to click on SIS. So it will kind of pre-compile the information that you can then manually upload to SIS. That's the manual way to do it. Probably mainly suited for reassessment, where you will have just a couple of parameters to complete and tweak. The other possibility is to use a more automated way, which is through SIS Connect, a tool that is created by the sRedList unit. You need special permission to access that, so you can check in the sRedList documentation how to get that permission. The idea is that based on some spreadsheets, you can create working sets that the sRedList unit will manually check and migrate to drafts in SIS. And those spreadsheets for the working set are exactly the one we provide you here with the assessment. So you can take those files, put them in SIS Connect, wait for the sRedList unit to check them and upload to SIS, and then all your countries of occurrence, your habitat preferences, your estimates, your justification will be automatically loaded to SIS. And then you can just go and edit and complete your assessment there. In order to minimize the work of the sRedList unit, if you are doing many first assessments and want to use SIS Connect, we ask you to use the merge zip function here, where you can select different zip files that were created on the sRedList platform, so the one I just created and some other species I compiled before, upload them, and it will return a single zip file, including those 3 assessments that you can then push to SIS Connect. I will now conclude. If you like the platform, which we really hope, it would be great if you can share the word, spread the word within your groups with the other assessors that you know, so that they can discover the platform. Also, we tested the platform, as Moreno was saying, in multiple ways and many times, but you will try it with species we didn't try, and maybe you will discover small bugs. So if so, we ask you to report it to us, either via the forum or via email, so that we can check if it's a real bug and solve it. And finally, we are interested in knowing how you use the platform. So we are always happy to receive some feedback. And in the future, we will also share a survey with you to understand how you use the platform and what else could be useful. Thanks a lot for your attention. It was great to present you the platform, and I hope you have some questions and that you will like discovering that tool.
Well, that was impressive, Victor. Thank you very much. I did my best to keep track of questions. There were many, indeed. A lot of congratulations for all your hard work and a few questions. So I did my best to answer some of them, and I believe your presentation answered already. So, for example, Jean-Paul was wondering whether you could do a country-level assessment for species occurring in a single country, but I think you covered that in the last part. So yes, the functionality is available. So one question from Kelsey, the first one that she did, she gave a couple of questions. The first one is, can you change the presence and origins code of the range map you create, and how can you do that?
So currently we cannot, for instance, calculate, no, sorry, I didn't get the question right. Currently we can't. We cannot do that on the platform. Everything will be coded present, 111, resident and native. And so you will have to do that in your GIS or in the spreadsheet afterwards.
Okay. Then there was another question from, sorry, keeping track a little bit, from Oscar, I believe. So he was asking whether you could do multiple species at the same time. Now, as far as I understand, but correct my answer if it's wrong, in order to do 2 species, you need to log twice on the platform. Is that correct?
Yeah. So you can work on them at the same time, like you can have two pages open, but there is no batch functionality for now. We hope to develop that in the future in order to say, okay, I have those 10 species, I want to see what the area of habitat is. But we made the platform so interactive that we ask many questions on the way, so it will require some work and some rethinking of the platform probably.
Yeah. So one more question from Kelsey, that's still open. So she said, other than habitat loss, can we account for other threats, disease, overharvesting, climate change? How can we do that with the platform?
We don't have that in the platform yet because we didn't see any methodology that is valid to do that. So Giordano Mancini developed one for climate change. We'll integrate the platform in the future. We are quite open when we will get funding to add some other. If you see like a great map of disease or a great methodology that would inform you on the disease spread of some species. So if you see something that is clearly lacking and would be helpful, we are happy to get the suggestion, but so far we didn't find anything convincing to map, especially those different threats.
All right. I'm sorry if I go quickly, but we have a few minutes left and a lot of questions. So I'm moving quickly to Jean first, I guess. So the question is, do you have a list of case study output from the examples that you showed with some of the species that have been used to test the platform? I guess they're asking whether there is a worked example that is published anywhere in the documentation.
So to answer the first part of the question, it has been used by testers, and if you look for the references in SIS, you will find some assessments using the sRedList platform. But we don't have an example where we show every step for one species. The reason is that some steps will be useful for some species, not so much for others. So in the video tutorials, for instance, I use a different species for each video, but it should be what will help you understand exactly what each step can do. Yeah.
So to answer to you, Jean, yes, the video will actually show you a fully worked example. Well, there is multiple videos with fully worked example of actual species.
Yeah.
Then one from Riley, I hope I'm pronouncing your name correctly, and was asking, it would be great if this could be developed further to include functionalities for the green status assessment. Now, it might be a little bit of a side question, but we discussed that, so maybe you can elaborate a little bit on this one if you want, Victor.
Yeah, so we have
green status assessment.
Sorry?
Can we do green status assessment?
So currently, we try to develop it to fit really the need of RedList assessments. Maybe there are some steps that could be useful for green status, but it's not developed for that currently. It would be maybe someday in the future something we can add. We have Rejita Chakaya in the group who mentioned that a couple of times. I think my vision so far is that the green status first need to build a bit more on the ground level to understand how it can be used in a more automated way. But at first we need to understand exactly what is the information that can be useful to green status.
Good point. Moving on to the next one. Sorry if I am bombarding you with questions, but there is quite a few. So question from Gert. How detailed is the habitat classification? So say you have a freshwater species living in a small forest stream. A selection of forest as habitat is too broad. Can you go deeper? Can you specify this information in the platform?
So on the area of habitat step, we use what is published, like what the science has said is valid. So what we do is that we use the IUCN habitat. So we don't go more detailed than the IUCN habitat level 1 and 2. And then the thing that is quite broad is the crosswalk from the IUCN habitat to the global land covers. So then it's also published. You can check it in the documentation. but maybe that will be a bit broad for the use you say. The best is to look at the map and see if it makes sense or if it's just too broad, in which case we don't have the global data to assess something finer. And for the freshwater area of habitat, I know that there are some people trying to develop a better methodology for that. Currently, it's mainly made for terrestrial species, so probably in the future we will have something more suited for freshwater species because they need something specific.
Yeah, and if I may quickly add, I remember the only real trouble we encountered in the past was one of the species we tested in one of our tests was a specialist of a very specific localized type of habitat. It was a butterfly of the rocky outcrops. So you imagine a species that can live in tiny, teeny patches of, maybe 10 square meters or something here and there in a bigger landscape. We cannot go to that level with such a generalized tool. So for that sort of species, it's unlikely that a tool like this will give you everything you need. It can give you, a first broader understanding of the habitat, but then you need to probably work and refine that information. That's not likely what's going to happen. All right. Question from Mario. Criterion B is probably the most used. Can you implement tools that allow visualization of locations and fragmentation for criterion B?
Fragmentation, we have it in the platform. I didn't show you because it's a bit of a technical step where you need to understand stuff and we had limited time, but you can, based on the area of habitat, if you are happy with the map of area of habitat, you can calculate severe fragmentation. For the number of locations, it's too tricky. Like the definition of the location that depends on the threats. So it could be super big if it's climate change that is threatening your species, or very localized if it's pollution or whatever. We cannot map that in an automated way. It would be wrong 99% of the time, I think. So currently we don't have that. If you think of one way that would be very useful for one threat or whatever, we are happy to hear about it.
We can do a few things, not everything. I guess that's the point.
Yeah. We are happy to add it somewhere in the wish list, and then based on funding and possibilities, we will add some stuff.
So a question from Hugo. I'm not sure if I'm interpreting that correctly, but I believe it refers to the local scale application, and is asking what the minimum resolution of GIS sources to use SIS. Is it possible to use it at local level, for example, within provinces or departments? So I don't know whether that refers to local scale assessment, like assessment within a single province. In principle, I guess we could do it as long as the province is big enough for the resolution of standard land use maps. Is that right? What's the resolution of your land use product that you just
the CCI is 300 by 300 meters. So for some species that are quite localized, it works well. And then you will have the known area of occupancy and the upper bound with area of habitat that are quite comparable. And so it's quite useful for localized species. But then, of course, if it's really, really tiny, it won't make a lot of information, I guess.
And my suggestion is, if you want to go that deep, that local of a scale, you always need to check that the habitat map, which we have globally available, makes sense in your particular location. Those maps are, broadly speaking, those are good over large extents, but over specific localized extent, they might or might not be good. So please double check. If you want to go very local, double check that the map, in your specific location makes sense. Of course, the RedList assessor is the only expert of that, so we cannot provide any prior information on how deep and how local you can go. One from Oscar. How can we properly cite this tool in SIS? I believe this is an easy answer, isn't it?
Yeah, but important, I should have mentioned. So on the homepage of the platform, you have a citation that is suggested. And also, I didn't show that, but in the output, if you're using SIS Connect, you have a file with the references that will include the sRedList platform, some packages that we use, and also all the references from the occurrence record. If you started from occurrence records, that you probably have to do some tweaks manually, but the references are here one line for each, and you can upload that in SIS directly. So you don't have to go in SIS, put every reference of your occurrence records. Thanks for the question.
Great. And one from Jairo. It could be awesome if experts could load some shapefile with more precise local, regional, suitable habitat assessment so that user can use the skills of the platform for the calculation. So in other words, can an expert upload their own habitat suitability map? I guess that was the question, if I get it correctly.
The habitat suitability or the shapefile?
I think is talking about habitat suitability, because it says more precise local regional suitable habitat assessment. So not the range, but the habitat, I think.
Currently,
if I'm wrong, Jairo.
Currently, we don't have that possible. You cannot say, I have a good land cover map of Brazil, so I will upload that and check what happens there for my species. But maybe something we can add in the future. It's quite tricky because it needs a lot of harmonization of the data input. But it could be, yeah, it's somewhere in the wish list already.
Yeah, I guess the main issue there is as long as the land cover classes are compatible with the global standardized land cover classes and with the IUCN habitat classification scheme, then it's in theory something we can get to at some point. But as long as the classes are compatible. All right, it doesn't look like there is more question. Hopefully I didn't miss any. We're exactly on time, so maybe if there is one last burning question for Victor, we will do it. Otherwise, we will just thank Victor for the brilliant presentation and for answering all the questions. And once again, maybe if there is no more question, Victor, can you remind us when is the platform gonna be online and how do we?
Yeah, so it's gonna be online tomorrow. We still have some final testing to do, so sometimes in the day, maybe more in the afternoon, so that we are sure we're releasing something that works perfectly, we hope.
Yeah, but that would be afternoon Central European time, which is gonna be morning for the Americas, for example.
Yeah.
Is that right?
Yeah.
Fantastic. Well, I don't see any more question. No, sorry, there is one more. Sorry, Parna. Plot outcrops, we've been discussing the challenges in mapping outcrops. Oh, look at that. For every problem, there is a solution. So it looks like our butterfly example might have a solution from the website of our sharing. Well, thanks for that. So I would say thanks a lot to everyone participating. Really, please do use the platform. Again, it's going to take a little bit of a learning curve at the beginning, but it's not going to last long. And over the long term, or even the medium term, I would say, you will see yourself saving a lot of time, in my opinion. That's the experience we've had testing the platform so far. So thanks once again for the brilliant work, Victor. And maybe you can say a few final words, and then well,
yeah, thank you everyone
Thank you
everyone for attending. And as Moreno was saying, we are looking forward to see how you use the platform. So also feel free to write us on the forum and share your experience of the platform. It will be super helpful.
Fantastic. Yeah, we've done this one for you. Use it. Let us know what you think. All right. Bye-bye, everyone. Goodbye.
Thanks. Bye.